Today, we're diving deep into a muscle condition known as myofibrillar myopathy, or MFM. This condition was once grouped under the term type 2 polysaccharide storage myopathy, but it is now recognized as a distinct myopathy. Myofibrillar myopathy is not a glycogen storage disease. This podcast will explore what myofibrillar myopathy is, how it affects horses, and what can be done to manage it, drawing from a recent veterinary publication. Understanding skeletal muscle. To understand myofibrillar myopathy, it's important to understand the basics of skeletal muscle. Muscle fibers are strategically oriented to generate power and motion while maintaining structural integrity. Within each muscle fiber are myofibrils, which contain myofilaments. These myofilaments, mainly actin and myosin, are arranged into contractile units called sarcomeres. The M line anchors myosin filament to the center of the sarcomere, while actin is anchored by Z discs at the ends. Desmin, an intermediate filament, threads through the Z disc, aligning parallel myofibrils and giving skeletal muscle its striated appearance. Desmin also anchors myofibrils to the cell membrane and positions organelles like mitochondria. The Z disc acts as a mechanosensor and mechanotransducer, upregulating gene transcription in response to training and creating exercise adaptations. Skeletal muscle has 3 contractile fiber types based on a myosin heavy chain. Type 1 fibers, prevalent in postural muscles, contract slowly and rely heavily on oxidative metabolism in mitochondria. Type 2 fibers are fast twitch fibers, making up a large portion of locomotor muscles, and have subtypes. Type 2A fibers have high oxidative and anaerobic capacity, while Type 2X fibers have low oxidative and high anaerobic capacity, especially in untrained horses. History of the disease. The identification of myofibrillar myopathy came after the discovery of the H309GGYS1 genetic mutation, which causes type 1 polysaccharide storage myopathy. Some horses showed abnormal polysaccharide accumulation but did not have this mutation. These horses were initially diagnosed with type 2 polysaccharide storage myopathy based on abnormal amylase, resistant glycogen, or small aggregates of amylase-sensitive glycogen. A subset of quarter horses with exertional rhabdomyolysis and type 2 polysaccharide storage myopathy were found to have a glycogen storage disease. However, Arabian and warm-blood horses diagnosed with type 2 polysaccharide storage myopathy did not have high glycogen concentrations in their muscle, suggesting another disorder. Discovery of myofibrillar myopathy. Further studies using electron microscopy and immunohistochemical stains revealed myofibrillar disarray and ectopic accumulation of the cytoskeletal protein desmin in muscle fibers of Arabian and warm-blood horses. This led to renaming type 2 polysaccharide storage myopathy in these breeds to myofibrillar myopathy, with immunohistochemical staining for desmin used for diagnosis. The previous diagnosis of type 2 polysaccharide storage myopathy in Arabians with myofibrillar myopathy was likely due to small glycogen pools forming between disrupted myofibrils, creating the false appearance of cytoplasmic glycogen aggregates. Arabians and warm-bloods with myofibrillar myopathy have different clinical presentations and molecular signatures, including altered mitochondrial complex 1 and a propensity for low cysteine-based antioxidants. Arabians often present with high serum muscle enzymes and exertional rhabdomyolysis, while warm-bloods present with exercise intolerance and normal serum muscle enzymes. Myofibrillar myopathy in Arabians. Myofibrillar myopathy in Arabians, or MFM-WB, causes intermittent episodes of exertional rhabdomyolysis, often in older endurance horses at the end of prolonged rides. Clinical signs may also appear about 5 miles into a ride after a 2-week break. Horses exhibit muscle stiffness and pain, though less pronounced than in other causes of exertional rhabdomyolysis. Serum creatine kinase activity can be markedly elevated, and myoglobinuria may be evident. Without treatment, clinical signs may become more frequent with less exertion. A diagnosis of MFM-WB is confirmed by correlating clinical signs, serum creatine kinase activity after an endurance ride, and histopathology in a muscle biopsy. Immunohistochemical staining for desmin is essential. Muscle biopsies may also show internalized myonuclei and, occasionally, amylase-resistant polysaccharide in a few fibers. Pathophysiology of MFM-ER. The heart rate and metabolic response to exercise are normal in MFM-ER horses, and muscle glycogen concentrations are similar to controls. However, transcriptomic and proteomic analyses reveal alterations in cysteine synthesis, altered expression of complex 1 subunits in the mitochondrial respiratory chain, and lower content of the cysteine-based antioxidant peroxidoxin 6. Arabian endurance horses tend to oxidize fatty acids, producing reactive oxygen species during aerobic exercise via complex 1. The combination of a lack of cysteine-based antioxidants and a highly aerobic metabolic phenotype is thought to promote oxidative stress, oxidation, and aggregation of structural proteins like desmin, potentially oxidizing the ryanodine receptor, leading to exertional rhabdomyolysis. Genetic testing for MFM and type 2 polysaccharide storage myopathy is available, but research indicates that these tests can produce false positives and negatives. Therefore, muscle biopsy remains the gold standard for diagnosis. Now, let's discuss managing myofibrillar myopathy in Arabians.
Managing MFM-ER. Since MFM-ER is not a glycogen storage disease, a low non-structural carbohydrate diet is not scientifically supported. Dietary management should include moderate levels of non-structural carbohydrates and fat, with higher levels of protein. The most absorbable form of cysteine is N-acetylcysteine, which has been shown to diminish desmin aggregation in a vertebrate MFM disease model. Supplementation with N-acetylcysteine can increase muscle glutathione, an antioxidant. A commercial supplement containing N-acetylcysteine and other amino acids has been developed for MFM horses and has shown to decrease clinical signs of rhabdomyolysis. Coenzyme Q10 is another antioxidant that may benefit endurance horses by enhancing flux through complex 2 rather than complex 1, the complex generating the most reactive oxygen species. Selenium and vitamin E levels should also be tested and supplemented if deficient. Regular daily exercise is important, as time off can increase the risk for exertional rhabdomyolysis. After endurance rides, some light exercise is recommended rather than complete rest. Myofibrillar myopathy in Warmbloods. Desmin aggregates have also been identified in warmblood horses. However, the underlying cause and clinical signs differ from MFM-ER. Warmbloods with myofibrillar myopathy rarely develop elevated serum creatine kinase or exertional rhabdomyolysis. They often develop persistent exercise intolerance, mild shifting hindlimb lameness, and mild to moderate muscle atrophy. Clinical signs of MFM-WB. Warmblood horses with MFM-WB often show promise as young horses, but develop an insidious onset of exercise intolerance around 6 to 8 years of age. This is characterized by a lack of stamina, unwillingness to go forward, inability to collect abnormal canter transitions, and an inability to sustain a normal canter. Muscle atrophy may be present, affecting the top line and hindquarters. Horses may show signs of stiffness and muscle pain upon palpation. Although owners may report tying up, serum creatine kinase activity is usually normal. A mild shifting hindlimb lameness and a higher prevalence of gastric ulceration are also common. Diagnosis of MFM-WB. Because the clinical signs of MFM-WB are nonspecific, other causes of pain or exercise intolerance should be ruled out first. This includes saddle fit, training issues, equine asthma, gastric ulcers, and orthopedic problems. Equine neuroaxonal dystrophy should also be considered. Muscle biopsies are required to establish a diagnosis of MFM-WB in horses 8 years or older. Gluteal or semimembranosus biopsies contain scattered myofibers with desmin aggregates. Desmin aggregates may not be apparent in younger horses, so a treatment trial may be necessary to diagnose MFM-WB in horses less than 8 years old. A clinical response should be seen within 4 weeks of starting treatment. If not, another diagnosis should be pursued. Genetic variants associated with MFM in humans have not been found in warmbloods with MFM-WB. Commercial genetic tests for MFM are available but can produce false positives and negatives. Therefore, these tests are not recommended. Pathophysiology of MFM-WB. In MFM-WB, fast-twitch oxidative type 2A muscle fibers contain scattered areas of myofibrillar disarray, Z-disc streaming, disruption of the Z-discs, and ectopic accumulation of Z-disc proteins like desmin. The Z-disc contains hundreds of proteins involved in mechanosignaling and mechanotransduction. During mechanosignaling, Tension from muscle contraction stimulates specific Z-disc-associated proteins to migrate to the nucleus, activating transcription of genes needed for training adaptations. Proteomic and transcriptomic analysis reveals aberrant expression of proteins involved in mechanotransduction, decreased mitochondrial complex 1 expression, and a pro-oxidative cellular environment in affected horses' muscle. The gene with the highest expression in MFM-WB is CHAC1, which degrades the antioxidant glutathione. Thus, MFM in Warmbloods appears to arise from aberrant gene and protein expression affecting the alignment of contractile proteins, mitochondrial proteins that generate reactive oxygen species, and molecular training adaptations. MFM is likely caused by complex interactions between diet, environment, and multiple genes. Managing MFM-WB. Management of MFM requires altering diet and exercise regimes. Dietary principles include hay with 12% to 17% non-structural carbohydrates and a concentrate with moderate levels of non-structural carbohydrates, lower fat, and higher protein. Adjustments should be made for horses with obesity, metabolic syndrome, or laminitis. Whey-based protein supplements are recommended because they are rich in cysteine. Branched-chain amino acids such as leucine may also be beneficial. A commercial supplement containing N-acetylcysteine and branched-chain amino acids has improved exercise tolerance in MFM horses. Coenzyme Q10 can be provided as an additional antioxidant to enhance the production of NADH, which reduces oxidized glutathione. N-acetylcysteine and coenzyme Q10 have been shown to increase glutathione in muscle following exercise. Unlike exertional rhabdomyolysis, warmbloods with MFM require days off training to recover. 3 days of work followed by 2 days of rest often works well. A warm-up on the lunge line can improve ridden work. Hill work, cavaletti small jumps, and core strengthening exercises can be used to build strength. Daily turnout is also recommended. Behavioral issues may require groundwork and the help of a professional trainer. Conclusion. In summary, research has revealed several muscle disorders previously grouped under the term type 2 polysaccharide storage myopathy. These include type 2 polysaccharide storage myopathy with exertional rhabdomyolysis in quarter horses, myofibrillar myopathy with exertional rhabdomyolysis in Arabians, and myofibrillar myopathy in warmbloods. Myofibrillar myopathy in Arabians and warmbloods both involve desmin aggregates in muscle and evidence of oxidative stress with a lack of cysteine-based antioxidants. Myofibrillar myopathy with exertional rhabdomyolysis appears largely due to oxidative stress associated with a lack of cysteine-based antioxidants. Myofibrillar myopathy in warmbloods appears more complex, also involving aberrant training adaptations. From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.